Okay, let's begin. Module 3. Let me turn the speaker thing on. Module 3, it's a combination of repair, and then all of the rest of it goes into transcriptional mechanisms in bacteria. And then module 4 will be gene expression in eukaryotes. So we're getting into some of my favorite topics. Transcriptional expression is something I've been interested in for maybe 40 years now. So hang on. But first, repair. Is repair important? I mean, you eat right, get plenty of sleep. You don't have to worry about DNA repair, do you? Every time you walk outside in the sun, your DNA is getting damaged by UV, and it's getting repaired, fortunately. Every time I walk out in the sun, my DNA is getting damaged, but sometimes it's not getting repaired. I got big bumps all over my head because I don't wear a hat. Those cells are permanently, forever mutated. How about genes inside of you? Are mutations happening all the time? Oh yeah. It's just they're not happening in genes that happen to be vital for that particular tissue, so you don't know about them. You've got stuff happening all the time. The food you eat, with all those free radicals and stuff, that's really damaging. You need something to maintain your genome integrity. But, you know, these cells here, they're destined for glory, right? They're not going to last forever. Dust to dust. So the organism and evolution doesn't care so much about anything but the germline. And every time you have a germline involved event, it's pretty much the whole genome is tested. Like, let's fire it up and check everything, make it works. Development, going from embryo to full great big person, is the hardest thing the genome has to do. The coordination, the synchrony, the perfection and timing. If it's not right, it has a bad outcome. Your genomes are pre-checked, run around the block. I mean, they're raced around the block to make sure mutations have not accumulated, because they're accumulating everywhere. The bases themselves are not stable. Oh, don't tell me that. They decay naturally without any kind of radiation or anything. They just have a certain half-life. Some of the bases are more unstable than others. They are breaking down right now as we speak. Okay, maybe I have your attention. Repair is important. Let's see how it's done. And I'm going to change out this title slide to make it more relevant to your current edition of the book, but otherwise it's pretty up to date. More than 130 genes in us are devoted in one way or another to the process of genome maintenance and repair. And you can see here sort of a listing. I don't require that you expect you to know all these. You'll become familiar with these as we go along. But you see there are a variety of mechanisms and processes in place that handle nothing but repair. So first of all, we're going to talk about the kinds, the types of repair, and then we'll switch into particular pathways of repair where you'll have to know these names of proteins and activities and stuff, and those things will end up on a key list, keywords list that I hope to get out on a more timely basis for Module 3. So let's look at the types. There are, 4 main categories. Direct reversal. If something is broken, fix it. Don't cut it out and get a new part like all, auto repair and everything. They just say, they never fix anything. They just replace stuff. Nature does that mostly also, but there is one example where if there's a mistake, the mistake is fixed directly. And it's notable. We're going to see what that is in a minute. But much of the repair involves just cutting out the damaged area, identifying the damage, and then cutting it out, and then synthesizing a new strand over 3 or 4 bases, 1 base, 1,000 bases, 9,000 bases. Varies on the system. But replace much more than actually the damaged component. So excision, remove and replace. And of course, what do you can excise the base, the entire nucleotide, or you can go into a mismatch and correct that. So this is a major category here, excision. Then you have recombination repair. After this Module 2 that there's a little repair that needs to be done. When transposons move, there's a little repair, and recombination, the reactions are, in some sense, are repaired. Displacement strand goes up there and you have to kind of fill in the gap and stuff. So recombination related repair. And then non-homologous end joining, just because it's so sloppy, you end up having to repair as a consequence. What is non-homologous end joining used for in us? The VJ joining. And then synthesis of replacement DNA. I don't know how that fits in as a repair, but anyway, the 4 there are understandable. Types of damage. So let's look first at the distinction between a category of damages that are so severe in terms of DNA synthesis that they stop the enzyme. And then we'll look at the other category that, while it's a mutation, or it can lead to a permanent mutation, it's not enough to stop DNA polymerase or RNA polymerase. So let's see what they are. Single base changes, let's go through the whole thing here. So single base change affects sequence but do not interfere with transcription or replication. So these are not severe enough in terms of the enzymology to stop a polymerase. And that would be a mismatch, a misincorporation, a deamination. More about deaminations to come. This is the unstable aspect of our bases. The ones with amine groups, they tend to just decay on their own. They have a certain half-life of stability. Misincorporation, deaminations, a mismatch that persists only until the next replication, one copy is permanently mutated. So that's a little bit cryptic, but what we're saying here is if the mutation or damage is not enough to stop the polymerase, you go, cool, no big deal, didn't stop my polymerase, ha ha ha. But that turns out to be the most dangerous kind. Why would that be? If you didn't stop the DNA polymerase, that means the polymerase is going to copy that mistake. And now you have that mutation forever. Becomes permanently incorporated. So what on the surface seems no big deal, it is a big deal if it's not repaired before replication. Because if it's still there after replication, you're stuck with it. There's no way to ever recognize that it is a mutation. Okay. This is a cheerful message that I'm giving you guys, okay? Single base change, so misincorporation, deamination. How about the second category. These are an impediment to transcription and to DNA replication, and all of a sudden we go, yay, it's time somebody put a stop to this crazy damage. The polymerase itself can't proceed, so now you have a chance to actually recognize that it is damaged so that you can fix it. So examples of these would be thiamine dimers by UV on the University of Florida campus, walking between classes. That's enough to do it. Bam. Alkylation of a G. Alkylation's adding a methyl group to a G. You can add a methyl to a C. That will cause a mismatch, but a methyl to a G, because of where it's added, causes incorrect base pairing. And it causes a bulge in the duplex, and it'll actually stop transcription and DNA synthesis. A single strand nick. What's a nick? In the phosphodiester backbone. If you just break that phosphodiester backbone, don't remove a base, just snip the backbone on one of the strands. That completely stops transcription and DNA replication. A nick. And the last, of course, if you remove the entire base but leave the phosphodiester backbone intact, that also will stop replication and transcription. There's no base there to pair with, so it stops. Thymidine dimers. Okay, so the two kinds, sometimes on exams, I will have a list of things and say, which of these would stop DNA replication? By that I mean stop the enzyme. So you're going to look for thiamine dimers, alkylation of a G, single strand nick, removal of a base. It causes mutation because it does not stop replication. Thiamine dimer. We've thrown this term around a lot. What is it actually? It's two thiamines that are adjacent on the same strand. Those big polar rings you see there, those cross-link, chemically cross-link with each other side by side in the presence of high energy radiation, UV radiation. Cross-links them to form a thiamine dimer, and here you see the cross-linking. This event is enough to make it so that, you know, this is normally paired up with an A. If you had a polymerase that came running along here, it would not be able to flip the template one base at a time so it could be properly read. The two would be attached to each other and it wouldn't fit in the active site properly. So you would stop the enzyme, both the DNA polymerase and the RNA polymerase, for that reason. And it also causes a distortion that can be detected by scanning bots that we have crawling our genomes all the time, looking for mistakes. This kind of mutation that's induced by UV, this is the only one that can be reversed. You know, we said that in the types, we can actually, not we, but there are repair enzymes that can go in and just break the covalent bonds. Light energy created these troublesome bonds, and it turns out the enzyme is called photolyase. Lyase for cut. Photo means it uses the energy of light to repair what energy of light did. So a photolyase is an enzyme that actually, it doesn't cut anything out. It's the one example that fixes the problem. This is found widely in plants, widely in bacteria, even bacteria that live where the sun doesn't shine. Bacteria that are deep in the earth. bacteria deep in there if you dig them up. You know, half a football field under the ground, dig it up, and you can isolate photolysins. And it's conserved. Very strange. This is methylation or alkylation of the G. So the thymine dimer could stop replication and transcription. Alkylation of bases, and this is the specific example, methylation of a guanine disrupts the base pairing such that it causes a distortion in the double-strand DNA and can be recognized as something that needs to be fixed. And because it can't base pair properly, the DNA polymerase cannot proceed through it. Depurination. This is a very important one because this depurination, well, it's deamination is important, but depurination is important also. Notice what it is. This is the ribose sugar, deoxyribose sugar. This is the backbone. And this is the base, and it's adenine or guanine. They are purines. And this thing is pretty complicated and not that stable. Purines are fragile. They just break down with time whether you expose them to radiation or chemical mutagenesis or not. You get a loss of this entire base portion, and now you do have something that definitely will stop an enzyme, replication, transcription. And here we get to the one that is also very interesting. This is called deamination. The amine group on cytosine, here you see cytosine, and this amine group here is not very stable. Who decided to use C as one of our building blocks of the genetic code? This constantly has to be fixed. It decays, it's decaying right now as we speak. I heard another one go. All over there where you're sitting, these things are going off and having to be fixed. Notice what happens. A C, all it needs to do is lose this fragile amine group and it turns into a U. Uh-oh. It turns into a U. Well, U, that's kind of friendly, right? It's the base. No, because this now makes it hard to distinguish between, is that a bad U or is that a good U? That's like your evil twin. Is that the bad U or the good? Anyway, so a U in DNA, there are lots of systems that recognize that, right? Because that's just the nature, the difference between RNA and DNA. And every enzyme of repair knows if I'm working on DNA, I don't need a U sitting there. So thank goodness. That is one, if it's a U, no problem. There are lots of uracil glycosylases that go and say, you know, this U, because what is this going to be paired with? It used to be a C. So its base pairing partner is a G. But if this thing just falls off, becomes a U, now it's a UG base pair. It's a perfect U. It's a perfect G, and yet by default, the cell has enzymes in place that go, I'm sorry, but U's are not allowed here. This is DNA. DNA only. So what would be the other time you would see a U? Well, since RNA in the primer puts a U in across from an A, doesn't it? And then it gets switched out as a T. But here, the U is getting switched back out as a C. Whoa, that's tricky. So when you do the normal repair, you'd say, we got U's in here. They are obviously paired with A's. They need to be changed to T's. But if you're a U, is derived through this deamination pathway, it'll be sitting across from a G. If a U is sitting across from a G, it's repaired to a C. It's not turned into a T. Thank goodness. Okay. Uracil excised and it placed with cytosine if its partner is a G. This is such a frequent event that enzymes are in place by default without even thinking about it. They go, UG, I know it's not the G that's the problem, it's the U, and it shouldn't be a T, it's got to be a C. Cuts it out, replaces it. Notice another kind of deamination. If your C is methylated, and remember, C's are methylated all the time, right? Yeah, maybe you don't know that yet, but C's are methylated all the time, right? It's a common way to epigenetically deactivate DNA. It's a favorite base to put a C on. Now when you do that, there are pluses and minuses, because if the amine group decays off of 5-methyl-C, what you're left with is not U, but you're left with T. T in DNA, that's allowed. So how do you know it's a mistake? Well, this particular T is going to be paired up with a G. So if you're writing the rules about how to make life work, you're going to go, okay, a T and a G, I'm going to just guess that it's not the G that needs to be corrected to an A. It's going to be the T that needs to be corrected to a C. There's a bias in the repair system to make sure when you have mismatches, either a C or a T across from a G, you always assume the G is correct. Because this deamination process can lead to a CG or a TG quite easily. So remember, deamination of C gives you uracil. Deamination of 5-methyl-C gives you thymine. Thymine, of course, I mean, that's could lead to mutation because it looks like a normal part of DNA. Okay, so that's the kinds of mutations described in terms of whether or not they stop the enzymology. They stop DNA replication or transcription. Let's look at another way to characterize the systems. And that's the kind of system. Before we get into the specific examples, well, here's a specific example. Before we get into really deep into the weeds of the specific examples, let's look at the general types. And we've talked about this a little bit. Direct repair and then pretty much everything else. Excision repair. Direct repair, the one example, photolyse, it's the PHR gene, in E. coli, that's what it's called. It uses, let's see, reactivity is rare but important in plants, light activated enzymes, in animals with the exception of placental mammals. I think that's what my mother was. Oh dear. Let's see. In animals with the exception of placental mammals, I guess we've got this old-fashioned way, you excise it and repair, like all these other steps. Otherwise, it's quite common to have a photolyse in many, many, many life forms. What kind of excisions do? You can excise a nucleotide, you can excise a base, and here's some glycosylases, it's a good thing to remember, are involved in excising a damaged base is a uracil glycosylase. There are other kinds of glycosylases. We'll see this in a little more detail in a minute. Nucleotide excision repair removes a whole region, 2, 3, 4, 12 bases on either side of the damage. On either side of a thymine dimer, for example, nucleotides, whole nucleotides are removed, even though only one of them is damaged. Base excision repair just removes the one base that has the damage. Of course, it triggers other events, but the enzyme itself is just operating on one base. It doesn't remove a whole series at that step. Then mismatches. Mismatches that occur during replication are corrected by distinguishing between old and new strands. Look at this, cancer in humans and old bald-headed guys. Mutations in genes that are related to mismatch repair genes. We first characterized those in yeast, but mismatch repair has a big role in triggering when it goes wrong, or it gets tired, or it's just had enough after 183 years of life. It often can lead to cancer. Mismatches. And these mismatch repair systems, how would you repair a mismatch? You've got a G, it shines, it glistens in the sun, it's brand new, it's all fresh, it smells good. Next to a T, it also has that new car smell. They both are perfect bases. Which one is the mistake? How would you figure that out? Well, you start checking your list of basic assumptions. Mistake happened in the copying of the template. It's not going to be in the template, just by definition. Let's get some definitions straight. It's a mistake in the copy process. Certainly, the template was not wrong, right? Well, most mutations occur in that copying process, but not all, because the deamination, depurination, and all that can happen even in a template. So, but you could go a long way, especially if you could catch mutations right after replication, maybe that slip through the proofreading, and mutations do, and mismatches do even. It's all a probability game. If you could figure out which was the old strand and which was the new, you just make the assumption the new strand had the mistake. So now it becomes a problem of how do you tell which one is the new strand? Do you sneak up on it and see which one is cold and which one is still red hot because it's just made? Ooh, that's the new one. Mistake right here. How would you tell which is old and which is new?
Wouldn't the old one be hemimethylated?
Looking at modification pattern, hemimethylated, and that's the way E. coli does it. E. coli controls the frequency of DNA replication at ORAC based on this hemi versus fully methylated state at a particular site that serves no purpose other than to become fully methylated and then be hemimethylated. It's a powerful signal and it works very well. Just incorporate a methylation system that you can rely on, and you don't have to check the whole genome, you just go check at these particular sites that are deliberately there as a test of which is new strand and which is old strand. Yep, E. coli does that. And all the textbooks say that's how you do it. It turns out that only E. coli and some of the other gamma proteobacteria, I say that like I know what I'm talking about, I'm a botanist. Gamma proteobacteria, like Salmonella, E. coli, that's as long as the list I can make. They use this mismatch, this methylation, differential methylation, as a way to distinguish between an old strand and a new strand, but none of the rest of life uses that. Wow, how would you do it then? Everything else, in fact, that's a late addition, it appears. E. coli just figured this out after it became E. coli. It was like one thing it needed to do, get that mismatch repair thing going so it could be better than all of its friends. Everything else, including us, uses a whole nother system. It's sort of like, chasing a bus, and it's got the two big pipes coming out the back. That's DNA polymerase, lumbering along, smoke billowing out. And one strand is template, and out of one exhaust pipe is the new strand. If you always knew that the new strand came out of the right tailpipe, you could identify a new strand just by where it's exiting the enzyme. Possibly, if somebody told you ahead of time, it's always going to be on the right. But evolution is a great teacher, it just takes a while. So that turns out to be the way. What is it that clamps on the back of DNA polymerase? The clamp. Yeah, so the clamp seems to not just go fit on the back and go spinning around and have any kind of orientation. It clicks in at a certain configuration. And it seems that there might be topology, surface information that is there that could help a repair enzyme tell which is new strand, which is old strand. There's got to be something because that's the only clue we have. And it turns out these mismatch repair systems tend to work in tandem with DNA polymerases that have a clamp. And you'll see this in a minute where you'd think that repair would be done by DNA polymerase 1. That's the repair enzyme. And all of a sudden you've got Pol 3 doing a repair thing. There's probably a reason, because it has a clamp, and that clamp conveys information as to which is the new strand, which is the old strand. And none of this is in either of those textbooks. It's in the literature, but only you guys know. The MCAT people don't know it, so you might as well forget it. Oh, I digress. Let's continue here. Okay, photolysis uses the energy to break the cross-direct repair. How many times am I going to say this? This is 3 times so far. Light energy itself breaks the bonds and it's cured. Excision repair replaces more than just the damaged nucleotide or base. You get several or many bases are removed, and then you get replacement synthesis to fill in the gap that's created. Nucleotide excision repair has distinct stages. It's recognition of the damage, excision, or incision, cleaves on both sides. You see incision here on either side, although it's only very close on one of the sides here. It varies on the system. Sometimes it can be 3 or 4 on one side and 7 or so on the other, but in this example they're shown. But excision is the cutting, incision is the cutting, excision is removal, and sometimes this involves a helicase, and then you get synthesis, and a lot of times this is done by DNA Pol 1. Then ligase is the final step that seals the nick. So damage recognition, incision, which is cutting, excision, which is removal, synthesis, then ligation. Steps. Recognition, usually it's a distortion of the double strand. Incision, excision. Excision can be chewing away 5' to 3', exonuclease. It's often aided by a helicase. It helps unwind as it cuts. What is this? This is the same exonuclease activity that you see in Nick translation, right? It's just on its own separate, self-contained enzyme as a part of the repair. And then synthesis is usually Pol 1 replaces the damaged region. We'll get to this one exception, Pol 3, and because it's an exception, it usually ends up on exam 3, Pol 3. is used for mismatch repair. This mismatch repair, is the tricky one where you have to distinguish which strand is new and which strand is old. And then the last step is ligation seals the neck. Okay, so let's start with base excision repair, then we'll get to nucleotide excision repair, then mismatch. So here you see a deamination, and you get a regular C being deaminated to a regular U. Just lost its amine group here. And reverse by placing the U with a C. And I think this is largely what we've already said. Uracil glycosylase. These glycosylases are always a part of a base excision repair. Additional type recombination retrieval. We've talked about this just a little bit. We'll talk about it some more, mostly in bacteria. Non-homologous end joining, we've talked about that. Quality control, we haven't talked about the last one. It turns out, remember we said that DNA polymerase II was used to restart synthesis in bacteria, Pol2, DNA polymerase II, to restart at lesions. But then I also said polymerases 4 and 5 were used to restart at lesions. So what am I talking about? Pol2 is under normal mutagen conditions, where it's kind of the normal kind of base repair. The cell is not particularly stressed or anything. But this other group down here, tolerant systems, polymerases 4 and 5 are heavy duty. If you're under severe mutagenic attack, what you want to do is repair, but accept a lot of mistakes. What? Error prone. This is deliberately error prone. It's not very high fidelity. It actually puts in wrong bases on purpose. This is dangerous under normal conditions. So these error-prone polymerases are highly regulated. These are kept shut off except under extreme conditions. And the system that activates them is called SOS. Easy to remember. This ship, save our ship. Anybody know the Morse code for SOS? Some of you do. Dot, 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 dash, 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 dot, 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 dash, 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 dot, 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 dash, dash, dash. So you hear that now, you must feel obligated to notify somebody. That's SOS. Tolerance systems, they tolerate sloppy repairs. You see these TV ads where you can get this good repairman, they're all rated. This is where you look for the worst one and call him up. Come on over. Don't fix my plumbing. I want it to leak a little bit. Why would it do that? It wants to deliberately introduce mutations so they can have a chance to be selected. Maybe it can select itself. We can get natural selection to happen, and it can get out of this fix it's in. Maybe it can develop tolerance to this stressful condition it's in. So it's an evolutionary gamble. It knows that at best, 1 in a million is going to get a beneficial kind of mutation. It's going to be slim, but they hope somebody in the population here is going to make it onto the other side. So everybody becomes mutated. All your children, all your babies are mutated. And hope that somebody's little babies are going to be an improved strain of you. Because most of them are not. It's quite sad when they have to gather the community together and announce that they're going to have to start error prone. Tolerance. People, it's tolerance. I'm sorry. From now on, mutations. Welcome the mutations. Embrace the mutations. It's our only way out. It's like Planet of the Apes or something. I don't know. Okay. Tolerance. Error-prone system. That's the end of the audio file, part A1, 1A. Major pathways of repair. How many minutes? With 3. Okay, now we are in specific pathways. So we've gone from generalities to specific enzymes and pathways that you'll need to know for the exam. And I'll be your guide and I'll show you what's important. The UVR excision repair is really a fundamental starting place for most coverages of this topic. UV for ultraviolet, R for repair. The UV-induced repair, UV excision repair. Then our old friend, the DAM methylation-directed replication mismatch repair system. We've learned about how the DAM methylase is involved in the timing of DNA replication at OSC. Now we're going to see another role for this methylation system in identifying which is the new strand that needs to be corrected in a mismatch situation. And then we have another one of our old friends. Instead of REC-BC-D, you remember D was the fast helicase, like the rabbit and the tortoise. D ran fast at first until it met the chi sequence, and then everything stopped, and D was now very slow, and sometimes just dissociated from the REC-BC-D. REC-BC remained, and here REC-BC has some other partners involved in recombination repair pathways. So the UVR is the first, and we're going to need to stop after this slide, I think, but before we get into the particular enzymes, this is what you'll need to know. You'll need to know about short patch and long patch. Short patch is what happens normally. Long patch can involve, thousands of bases. It's very rare, and we're going to look at the difference. And the other thing, in terms of exam purposes, is you're going to need to know the subunits, the ends, the proteins as UVR, A, B, and C. And you're going to need to know the steps at which ATP is required. That's the concept that's important for exam 3. And I identify that in the subsequent slides here. Where is ATP involved? And it's not where you think it might be. When the UVRA recognizes the damage, but it needs ATP to leave the complex, so the next protein can come. So it's a little, I guess, counterintuitive that to leave, you think maybe it would need it to do something useful other than leave, but anyway. So we'll conclude today at this slide and keep marching through these things on Thursday. You got 5 more minutes? Then we'll not conclude on this slide, and we'll keep marching through these slides until 5 minutes is up. Misreading the hand signs down here. Okay, so we can get through this. Where are the two places when UVRA dissociates and where you actually make a cut, where BC cuts. But this doesn't mean much to you until you actually see some pictures, or some pictures are coming here. There's a UVRD, which is a helicase, and PAL1 is what is filling in, and ligase. And you're going, yada, yada, you're just reading your slides, which is true. When the picture comes, you'll see that makes more sense. When you're studying this, these will be kind of study points. ATP is required here. Of course, all helicases require ATP. And this is what I was talking about. 99% of the repair is short patch. Long patch repair is triggered under a very extreme conditions, the SOS, tolerance-prone situation where the cell's under heavy mutagenesis. Look how many bases are excised. 1,500 to 9,000 nucleotides. Although you got one thymine dimer, you're going to remove 9 kilobases. I wonder why that is. Maybe that's to allow some mistakes to occur as you try to repair that. I don't know. These are the players here. UVRB is always present, but it's in complex with a couple of different partners. UVRB first is in complex with UVRA, and the A subunit allows recognition of and most of the time it's a thymine dimer. A UV-induced mutation is normally a thymine dimer. It's not always. But the deformation of the DNA is recognized by UVRA, bound to B, and when the mutation has been recognized, then ATP is required for UVRA to leave, and now you get a replacement with an endonuclease, which is UVRC. UVRC is going to do the cutting, and it cuts asymmetrically, 7 bases on the 5' side of the damage and 3 or 4 nucleotides on the 3' side of the damage. Now, 7 plus 4 gives you 11. I don't know why this is like this, but it's in every textbook. It never adds up to 12, but details. And then after you've cut at 2 places on either side, then you have a excision, removal, and you remove in 2 ways. You can unwind it and degrade it, or you can simply go in and degrade it. There's a helicase that's often involved in unwinding it, and that's UVRD. UVRD, we're going to see, is also recruited in a mismatch repair. So you'll see a trading of proteins participating in more than one pathway. UVR excision repair system. Of course, helicases always require ATP. So the 3 steps are the leaving of UVRA after it recognizes the damage. I love that on exams because it's so counterintuitive. The nicking, the enzyme in order to cut, now that's doing work. You think maybe that might require ATP. That's UVRC is doing the cutting. And then when the helicase is here, UVRD, of course, that requires ATP. That's about it? Okay. And then how do you repair this once you've removed it? DNA Pol 1 fills in the gap, and then you seal the nick that's left by ligase. That's all there is to UVR excision repair. Not real complicated. What we'll cover next, I believe, is the one that's really beautiful, and that's mismatch repair. So, to be continued next time.